Okay, good morning, everyone. So the topic now will be interactions and network. So after the brief introduction about the multi-omics that Shintru did, we will talk now about complexes and protein-protein interactions. So you remember this pyramid, and you've seen it turn on the other side. But now to show you again, like this general idea, we start from the genome, then we have the transcriptome, we go to the proteome, and then we have, lastly, the interactome, that is this branch that comprehends everything, all the interactions that happen between proteins, RNA, protein and DNA, and like literally all the type of interaction that protein may have are studied by this branch here. As you can imagine, it's huge. I mean, it's one of the most complexest things to study. So in this particular talk today, we will just focus on protein-protein interactions. Unfortunately, because there is not enough time to talk about any type of interactions. But yeah. So what about protein-protein interactions? We all know that every protein, usually like most of the proteins, but kind of also every single protein needs to create a complex in order to be functional. Usually complexes are done with, could be the substrate, the thing that the protein is actively working on, but could be also complexes between different proteins in order to create an enzyme. Like, for example, you know, the ribosomes are like huge clusters of different proteins and RNA as well. So when we are interested into analyzing this kind of these complexes, the problem that we may have is that proteins in an experiment also tend to cluster together even when they are not supposed to do so. So we have specific binders, like as you can see from this very simple representation, that are proteins that are supposed to be there. They form the complex with that specific protein that we want to study. But then we have also unspecific binders, so other proteins that just attach to that one, just because of any kind of interaction that happened, just because they cluster together, also because it could be some issue, some error that happened during the sample preparation. And we don't want to analyze them because they are not interesting for the complex. And these are called unspecific binders. So the idea when you do protein-protein interactions is to get rid as much as you can of these unspecific binders. There are different ways. One could be, for example, purification, and then do identification. From what you see here, probably also you know better than I do, because these are more wet lab techniques. We have a protein A. We put here a binding protein, something that we know will bind surely with our protein of interest. And then we have here something that will attach as with the maximum affidability possible to protein A. Then we know that this site is the cleavage site, that then an enzyme that will cut this part will be used after in order to sequence only the protein of interest, the protein that will do the complex with the protein A, and will attach specifically to this calcium binding part. That then will be, we will wash again everything in order to remove any unspecific binder that attach to protein A, and we will selectively use this calcium binding protein to then select the specific binder. As you can see, usually you need to prepare your sample. You need to do several washes. You need to remove everything that was not is unspecific binder and you didn't want to analyze. And yeah, as you can imagine, is kind of complex and also requires time and money, this approach. But then something like 7 years ago, a new approach was introduced. I mean, not so new anymore, but the nice thing of this approach is that you don't care if you remove or not your unspecific binders. So it's called Affinity Enrichment Mass Spectrometry. So what you do, you have always something like an antibody here that will selectively attach to a bait. Bait is the protein that you want to sequence, and you want to cluster together with its specific binders. So what you do, you create two experiments, one with the bait and one without the bait, and you have to be sure that your specific binder here will select only the bait and not some all other stuff that may happen, that may be in the sample. And then what you expect is to find then your bait along with all the specific binders. But since you don't wash your sample, you will end up surely with also some unspecific binders that are here shown in gray. In your negative control, instead, you will have all the specific binders still floating in the medium. So after you will wash them, they will go away. But then the only thing that you will identify will be the unspecific binder. And the result you can see here, you expect that when you then take the sample and then perform proteolysis and then mass spectrometry and everything, here in the first condition, you will have an output from the mass spectrometer coming from all proteins, specific binders and unspecific binders. While in the negative control, all the specific binders will be washed out. And the only proteins that will remain will be the unspecific ones. As you can see here, we have signal only from the gray ones. And now you can imagine that you just use a volcano plot, and you will get rid of the unspecific binders. But we will see now this more in detail. So you have data coming from this specific paper, the paper in which Affinity Enrichment MS was introduced by Carlo et al. And yeah, more or less what they did is this. They used yeast that were marked with some specific proteins on this yeast were marked with GFP in order to then select these proteins specifically as baits. And then they took the lysis and they added a protein mixture. Then they performed what you saw before. They had their antibody that was supposed to select specifically the bait, B here shown. And this bait, as I said, was treated with a GFP protein. So there was a GFP domain there that will be that will attach selectively to the antibody. And then you see in I, written with I, the specific binders, and with U, the unspecific binders. They didn't care about the unspecific binder. They just perform a mild washing. In case the bait was not present, everything will be washed out. Then they digested all the proteins and continue then with the mass spectrometry. Just a few technical information if you want, they perform with orbit trap, but in whatever, we're not specifically interested into this, but then they use, thanks to bioinformatical analysis, they managed to get rid of the signal that was coming from unspecific binders. And then also, I mean, right now I'm showing you a brief introduction about interactomics, but then in also the USB stick that you have, you will find the data coming from this work, and then also in the practical part, there is, like, generally speaking, what was done into this paper, you will repeat what was done. And also, I will show you then you will have two different Perseus sessions if you want to repeat everything. One that will start from the pre-processing done, and one instead is the complete one. So if you want to then check every single step, you will have the complete Perseus session. It will be everything that I will show you later. Here, in case you're interested on how on some more specific information regarding this experiment, as I said, they used a GFP domain. They insert GFP into some specific part of the genome in order to create the bait. And in this specific case, they use as a negative control a histone protein, because they were expecting this histone protein not to create complexes with anything else, any other proteins that they were interested in And yeah, as you can see here, this specific domain was interested, and then the specific strain of yeast was used for the experiment. What they meant that with the volcano plot, you can recognize what is happening is like, for example, here, you see here that this is the signal coming from the mass spectrometer that's been analyzed after max quant run, and then this was done in Perseus. They analyze, they create a volcano plot, and they see the log fold change. You expect to see a positive log fold change if that protein attached to the bait. In this case, this is the control with the histone protein. As you can see, there are no proteins that are forming complexes with the histone. And the only enriched one is indeed the bait. In other case, here instead we can see that complex formed, and with specific bait. I don't remember if it was SK12 or 13, but one of these is the bait. And as you can see, then formed a complex with other similar proteins. And these were then had a positive log fold change. One thing usually that happens, if you see, when you do this kind of experiment and you see some proteins with negative log fold change, these usually are errors, because you don't expect a negative log fold change. I mean, usually you enrich for something, so it has positive log fold change, and the things that are unspecific binders, or the things that are just in the medium and are not interesting, they shouldn't change. So when you have a negative one, possibly is an error, or just a random error that happened. I mean, you cannot do anything for that, but another interesting thing they did is to reduce the kind of error and also to get more significant data, was instead of doing the log fold change between the situation with the bait and the experiment, the controls, they just decided to use as a control the entire amount of protein that was sequenced in every single situation. Because you expect that unspecific binders, you expect them to be similar in all your experimental conditions, because they are things that just attach there, and so they should be present in all the experiments. So what you can do, you can then plot your log fold change of the specific binders against this whole background noise. And it's called the complement, this. And you can see here with the t-test difference, we had something with volcano plot, the specific binder here, SET1, versus the control strain. The control strain, the histone. As you can see here, there were all the specific binders. This is the negative log fold change was the histone in this specific case. And yeah, they are significative, but they are very close to the unspecific binders. Instead, they tried to use more data. So let's say that now the background is 6 different other conditions. As you can see here, the unspecific binder remain there more or less, because they don't change. They are always there in every condition. While the specific ones now are their log fold change didn't change, but the p-value changed, because now we have more data, so the p-value is reduced. And then lastly, they went up to even 17 other experiments, like the entire data set they used. And we can see here that the p-value is so low now that the volcano plot is too small to show where actually they're supposed to be. Another way to further improve the data, actually, to further be sure about what they got, they also, with LFQ intensity, I forgot to mention that all these results were done with LFQ. They checked the different profile plots, and they thought, well, if we see something that has a similar profile, it means that it probably is a specific binder. Because otherwise, if binds unspecifically, we will see, for example, that protein enriched in one experiment, but definitely not in the other, because in the other something else attached there. So when they are binding unspecifically, the profile plot should be different in every condition. While here, they check the similarity, the correlation between the different profile plots in that specific, for that specific protein. And here, then, they further verified with the test-T difference for that specific bait and specific binder. And they checked also that the correlation in that single part of the profile plot was very high for these proteins, of course. But then also, if we get closer, you see here, if we get closer to the unspecific binders region, so with a lower p-value, then the correlation here is, as expected, very low. So these were general introduction about the data then we will use in the practical part that will follow further on. And how to represent, let's talk now quickly on how to represent networks or this kind of interaction. There are different ways. You can have an edge list. It's a simple table, nothing more than that. Usually network is used. And you can also go up to a multi-node network, but could be a risk. As you can see here, you can end up, when you have a lot of data, you can end up having something like this that is also called in a kind of negative way hairball, because you don't see anything from it. So let's start with the edge list. As I said, you just have your source. In this case, could be a bait, along with the target, the specific binders. Nothing. If you want, you can add more information on how significant was this interaction here, if there are any other targets also, but it's just a simple table at the end. But it's kind of boring type of representation, better to go for a network. And from here, you can see this is a very simple network. We have this central part here that we can call node. Usually, I mean, all of the circles are nodes, but usually the one in the central part of the network is the one that you use a bait. Or let's say the central part of the network is the protein that you actually describing all the interaction it does. And then we have edges that are these lines that could be from one side to the other. For example, this edge means that the interaction happened only between this protein and this one, but not vice versa. Here instead, both of them are interacting. This could be, for example, something like this could be, I don't know, for example, could be the substrate of a specific enzyme. So in that case, it's linear. It's directional, the interaction. And yeah, I mean, this is a bit nicer way to represent everything. And but how can we say if a network is well done or not? Here we have two different type of networks. Looking at them, we cannot say much more, but usually when we are describing biological effects, there is a general rule that works very well for networks. And is that the degree, network's degree. So the number of neighbors that every node has. When you have a random network, something like this one, this was a network that was just that is not representing any biological effect. It's just a random mix of network of edges and nodes. And as you can see, usually when you randomize something as kind of normal distribution, so you have on the top here something like, I don't know, this could be 2 or 3 interactions per node is the top, and then more you go here, they reduce, but also like there are few with just one interaction, with a lot of interaction here. While instead, when you do a scale-free network, so something that is more representative of a biological situation, what usually happens in a cell or whatever, you have a lot of nodes with a high degree. And then further on, they have way less degrees. But like the peak is with the highest number of degrees per node. Why? Because usually there is, sorry?
On the low.
On the, yeah, sorry, by mistake. On the low, sorry, on the low. And then you have few ones that have, and the difference here with the random is that you don't have like at the peak of 2 or 3, like in this case, you don't have so many nodes. You don't have so many nodes with 2 or 3 edges. That's what I wanted to say. So you have a lot of, like with just one, as you can see here, for example, there are all of these that have only one interaction, here 2, and all of this, because usually there is a central node that interacts with the many proteins. So this many protein will have only degree of 1. And then for their own, yeah, sorry, with the higher degree instead, that is reduced, and then there are very few proteins, and usually are your baits, those with a higher degree. If you're interested specifically in protein interactions, there are many databases and websites that will allow you to see what has been done so far. Like usually all the information about all the publications about interactions are also published into these databases. So you can just look for the protein that you're interested in and see which kind of information has been already published. I will show you here a simple example obtained from string. There is no reason why I've chosen string rather than the others. It's just my personal taste. I prefer string, but also all the others are all equally fine. I looked specifically here for this protein, this peroxyredoxin number 1. And the nice thing about, I like very much string also because there are not only the information about which kind of interactions is happening between cells, but depending on the colors, you have also information on how this interaction was discovered. Like if it was done through an experiment, so they have the real proof that the interaction happened, or as they found this interaction only through bioinformatical analysis. Like, for example, they know that there is this motif that usually interacts with that specific protein, and they found that motif repeated into another protein. And they say, probably an interaction may happen between these two, but there are no experiments that are showing 100% with 100% of certainty that this interaction is actually happening. And so you have also information about the structural information of the protein, and yeah, there are several information in string. I really strongly recommend you, when you're interested in a particular protein, just to look for it here in these databases, just to have a general overview of what it does, which are the interactors, and a comprehensive overview on the specific protein you're interested in. So now is the time for the practical part. And as I said, we will use the data set from this paper. And every step that has been done in this paper will be reproduced in Perseus. There are not so many steps, actually, very simple and linear workflow. As I said, there are 2 different sessions in the USB stick. This is the practical number 7. And you will have one session with the pre-processing done. We will open it now, that one, so we don't waste time during the pre-processing that you've done already yesterday. And also, if you want, then if you miss something here, don't worry, because you have also the complete session Perseus, where everything, you just can open that and went through that again. All right, so if you remember from yesterday, we just imported the matrix, simple matrix import. You have also the raw data in the USB stick if you want to start again from scratch. Then, as usual, contaminants and other proteins identified by side were removed, as usual. Then all the data was log transformed. We have here our nice data, but there are the missing value again. Every single row has been renamed in order to have, you see here, we have LFQ intensity for all B3. I mean, this could be useful when you go back to the experiment, but in this specific case, we're not interested in all those informations. So we're just interested in this name. This is the bait that we use and which replicate is. Then categorical annotation were added, so we can now the program, now Perseus knows which replicates are coming from the same experiment. So let's just take a look at the table now. So we have here the different baits. There are 4 replicates for every bait. So one is APC1, APC2. Here the baits are, if you remember from before, baits are those specific proteins that have been added the GFP in order to be selected. We have here then all the controls. They prepared several different controls with several different histone proteins marked with the GFP, and also each one of them has 4 replicates. Yeah, you have here all the LFQ values for all these experiments. They, as I said, they didn't care about what they were sequencing, they just sequenced everything. So these values may come from specific and non-specific binaries as well. And then here, as usual, the other information about your experiment. Filtering was done, and then at the end, data were imputed. The imputation was done with the normal imputation that you saw yesterday, replacing missing value from normal distribution. So now it's time to take a look at the data. So we'll just use an histogram. And let's see how many data were imputed. Well, here, this is a bit strange, but we can keep it. I mean, fortunately, there were 4 replicates. So also as a general rule, a very good thing is to do 4 replicates every time you do proteomics, because you're sure in case you lose something bad happens, at least you have still 3, and with still 3, you can still do statistics. But then we have this data here, this specific histone protein that was marked. Well, this looks very bad. So I will remove something that looked like that. I will definitely remove. And also we know that it's just one of the many controls there were, so we're not going to lose very much information. So in order to do that, wait, let me remember the name. So it was P histone 3, experimental replicate number 2, and technical replicate number 1. We can go here to rearrange, reorder, remove column. It was the number 2 and 1, so we can go here and 2 and 1, this one. So here on this part of the table, you have like all the information that you want to add. And if you want to add, you move them here. And here, there are all the information that you already have in your matrix. If you want to remove them, you then bring on the other side. That's it. And here also categorical, numerical number. Let's see now if I remove the correct one. It seems so, yes. So, of course, as you can see, there are also some other weird histograms here. That is up to you. If you don't like very much how the distribution is there and you want to have something more robust, then you may also remove them too. But I think we can continue with the statistics here with this data set. So how to do an interaction analysis? Well, there is a tool here in Perseus that is called the volcano plot. Sorry, Hawaii plot. a wave plot because there are many volcanoes. So what we have here, we have, this is as the same when you do a volcano plot. If you remember from yesterday, when Juan showed you how to prepare a volcano plot, nothing is changing here, except from one thing. You want to say which is your negative control. And here, we have bait, that was the grouping that was previously done. And you can decide between all these baits that have been used, which is your control. Like, for example, one thing that if you want to try at home, whatever, I can recommend you to do, you see, these are all the same control. One thing also to see how changes the result using more controls, more data coming from the unspecific binders. Something you can do is, like, for example, group together these controls or the histone protein that were marked, and see what happened if you just plot, I don't know, APC against one histone, against one replicate of histone, or then add several replicates, maybe all of them, all of the replicates, the biological replicates that has been done here with the histone, all of the 8 ones that were done. And you will see then that the significance will drastically increase more data you will have. But we're not going to do that. We're going to use the complement. And the complement is the information coming from the whole data set. So now we will plot the significant ones. We will plot one condition against the data coming, the LFQ results coming from all the others. So it means the information coming from the unspecific binder from all baits and all the experiments that were done. Here again, these are the default value for the volcano plot. We don't need to change anything there. We just press OK. Now it will take a bit of time because it will calculate it. Volcano for every single condition, a volcano plot that would be your bait against the complement. And then from this information, we will get which are the proteins that create a complex with the bait. Of course, if you're going to repeat something like this as an experiment, if you want to do affinity enrichment mass spectrometry, I think a good way to represent your data could be also this volcano plot, the Hawaii plot, actually, but that are nothing more but many volcano plots. But maybe you want to look for something nicer. That could be the network. And how to create a network from this information. Give a bit of time to the program to show now all the volcanoes. Here they are. As you can see, we have here the bait. Here it's a bit small, I understand, but you can zoom in, you can check, and if you're specifically interested in one, then you can make it larger here so we can see better. So yes, we have our bait protein with then all the possible complexes that it forms against the unspecific binders. They are all here, and here there are the possible proteins with positive log-fold change, so it means the proteins that may have formed a complex with them, with the bait. Something that I would like to point out that I think is nice because it shows that's exactly what we are expecting. We don't have any significant proteins for histones. Well, because we were not expecting for this specific condition to see any complexes. And that is good. It means that we are having a good experiment here. And all of the replicates with histones don't have any significant protein. Only those for different baits show some significance. Here. You can then select, again, as usual, which protein are these. Then you have the information about your bait here that also will have positive log-fold change too. But yeah.
Carlo, we have a question here.
Yes, please.
I noticed in your sort of theory slides and also on the examples given in the PDF file and on here that some of the volcano plots have two sort of lines sections. What does that mean? What does it mean if some proteins appear in between the two lines? Could you just explain that, please?
Those are two different p-values. The darker one, I mean, here you see better here actually. You see there is a dotted line and a continuous line. The continuous line is a more stringent p-value, 0.01, if I remember correctly. The other one is a less stringent, usual 0.05, no, 0.05, 0.5. And it is just a thing that you can use to see how sure you are about that. Lower the p-value is, better it will be. So like in this specific condition, instead, let's take this APC1. If you remember, was in the presentation, it was the one referring to the profile plots, where the different profile plots were compared to see how which was the correlation between them. Because here we have 4 very strong interactors, and we're kind of sure that they form a complex. But we have then other 4 here, and what can we say about them? Are they specific binders, or maybe they're just noise, unspecific binders that end up in such large quantity that we may think that they are specific binders? So yeah, these different lines allows you to see, like in this case, you see they are so these 2 proteins, and these are errors actually, but these 2 proteins here are so significant that we don't need to check between different p-values. Here they're all very close together, so then you see these 2 lines starting to get more distant. And yeah, what I would recommend you to do in this case, take those 4 proteins here and check the correlation with the profile plot. If they have a higher correlation, they could be specific binders, and maybe you have them here because they are very low expressed proteins. So the lower expression made them appear less significant. If you see then there the correlation with the profile plot is ridiculous, then probably they were only some unspecific binder in such large quantity in this experiment that they may have appeared as specific binders.
Thank you.
We have another question here.
Hi. Yeah,
Here.
I actually have 2 questions. First, the x-axis, is it t-test difference which that is depicted?
Yes.
Okay. And then if you go to APC2 sample, there's like a negative significant value. What does this mean? Like what exactly? Well,
in this case, you don't expect to find a negative significant one.
Exactly.
It's a random error, something that happened. But if you then specifically go here, let me see if it's correct. No, it's an error. In other graphs, it was the histone, because for some reason, the control group that was used as a histone then appeared there. But usually, you don't expect protein with a bait to be less expressed. Something has happened there. I will say that this is just an error that every time you do these experiments, also it's good to show that, but I will say it's that 0.1% that may always happen when you run statistics. That is an error, just a pure error.
I mean, you would expect as many of these on the wrong side as you have the FDR set, basically, right? So if you look into the 1% or 5% FDR interactors, then you would actually expect like 5% of them to be wrong, and so we'd also expect to be 5% on the wrong side to show up.
Okay, yeah, makes sense. Thank you.
Unless you have specific binding effects with the control, as you had with this histone as control that Carlo was showing, then you would expect even more on that side.
The second question?
Hi. Since there's so many replicates, right? I mean, each condition. So does the imputation necessary? Can we just leave the empty value there? We just don't do anything about the missing value. Just leave it there. Is that okay?
Well, yeah, I would say so. Yes. I mean, it shouldn't. No, it shouldn't. Doesn't give you an error, right? But I haven't. Honestly, I haven't tried with the non values, but it
Yeah,
shouldn't give
you wouldn't get an error, and it would work in principle. But I think you get better results if you do impute here, right? Because then you get like better like step profiles for the t-test, basically, if you use imputed values instead of just losing these values.
Okay.
And I will say also that the imputation, since in this case is working more for lower expressed proteins, is not going to change drastically information about your specific binders, because those they should be there every time. I mean, if you have a specific binder that ends up into a missing value in the condition that you're done, then probably that wasn't a specific binder.
So just make sure that you get a nice background to make the t-test against, basically, right? But it doesn't change much, as Carlo says, the actual binding proteins.
Yeah, one question to the y-axis. So the log p-value, this is in log 10, minus log 10 scale, right? Or can this
minus
be...
log 2. Yes, minus log 10. Minus log 10.
So, so the upper line with then 1% force discovery rate, shouldn't this run out at something like 1.3, which is then the, sorry, with 5%, the 5% line, the lower one, at 1.3, because this would correspond then to 5%?
No, because it's really the p-value that's plotted on the vertical, not the q-value. So it's really the p-value. And so that is then not, so the lines are created so that you get like FDR or Q value cutoff at like 5% or 1% indeed. But what is plotted on the line is actually the p-value.
I see.
Yeah, it was also a question that was asked yesterday about this thing. Is it Q value or P value? Here, the volcano plot is P value. Because otherwise, with a Q value, you will quench down. If you would plot that, you will quench down all the dots, and you will not see. The Q value is good when you want to find properly which things were really significant. When you then plot against FDR, when you then use the FDR, and you want to be really sure about the P values that you get.
Yeah, I guess you could do it also with Q values, and then like these what we call the curves, basically, they would become horizontal lines then, right, in these kind of plots. You could also do that. I think it doesn't look quite as good because so the way we calculate the Q value, so it's a little bit discrete. So actually, many of them also are, so that are very significant. They are then zero. They are really zero. And then if you log them, then they would be undefined again, or you would have to use some minimal threshold for those. So usually these, so first of all, as Carlo said, they become a little bit squeezed, but also they're not so pretty because of this discretization of the Q value.
So if there are more questions, then I would just ask you to wait 5 minutes, because I just want to show you last thing, and then we can have more questions. The thing I wanted to show you, because now we talked about networks, but this is not a network. And where is it? Here. So we have this button here that is called Create a Network from Significant Interactions. So what it does, Perseus now have all these interactions here, have all the volcano plots coming from every single bait with the significant proteins and everything, and will select every bait and will take only the significant ones, and will try to make an interaction network from this information. Use only enriched, yes. Remove bait edges, yes. So this is basic information about how to create the network. We will keep them as they are. And now we have here, you see, it changed color. And we have to go here now. So Perseus, we have right now we have two possible set of functions. We hope in the future then to make more. One set of functions work with matrices, the other one with networks. And here you see everything changes because you're working with networks and you have some functions. Again, you can do modification specific things. You can change annotation. It's very similar, but they are specifically suited for networks. And what is a network node from Perseus? That is this one. You see it's green. You can recognize also from the color and from the fact that it's called network. What it is two matrices actually. One that summarize everything in the network. We have the number of nodes, the number of edges, plus FDR. And here we have instead another set of two matrices. It's called Hawaii because we obtained it from Hawaii. But if you will import a network, then it will have the name of the imported network. So we have information here about nodes and information here about edges. Nodes are, there are here information about the baits, and it tells you which node is coming from, is a bait, and which node is instead an interaction. Then here you have the statistical information about your node and bait. And here on edges, it tells you which of these things here, which of these nodes in which we have the information should be connected together. When they should be connected together, there is a plus, and it has its own logful change with the significance. This is not the matrix, the table that I showed you before, the edge and node graphical representation. But it is very ugly, so you don't really see anything from it. So what we will do, it will be to use this function. It's called pull down network. So it will take all the information. Here it asks you which will be the node label. It's node, it's fine. Bait annotation, bait. And if you want, we can remove some parts of the network here, but let's keep everything. And what the program will do now is to summarize all this information from the network node into a graphical network. Here it is. So in the last few minutes that I have, I want to show you, are we seeing exactly what we saw before in the Hawaii plot? Should be, actually. So as you can see here, we have the gray parts that are baits. Some baits also interact with each other. And usually there are baits that are coming from proteins that, as you can see here, they have the similar names, so probably they make a complex together. And then in blue, we have all the interactors, all the significant interactors. Now we took all the significant ones, so also those that were, you remember the two lines in the volcano plot? Like, for example, here, also this has been selected for the network. If you want, you can go and select a more stringent p-value than for these. So you select only those that are very specific. But yeah, let's check. I don't know, this ZUO1 that has only this interaction. Let's go here. Let's check for that bait. Here it is. So what the network selected? This one. Not this one. Because this one, as I said, is an error coming from depending on the FDR that, as Jürgen mentioned before, the FDR that he used. The network selected this one. Is it for real? Let's see. Yes. It was only that one. And if you want, you can then go, if you have time, if you want to do, but this was only to show you how which of the nodes are selected to create the network. Then you can go through every node and see that is exactly one coming from that specific binder. Sorry, for the significant ones. So they are all these specific binders. And as you can see here in APC1 and APC2, We selected, indeed, as I said before, also those that were a bit less significant, still significant, but closer to this situation. You can recognize that because look how many we have, 4 and 4 for both of them, and they were exactly the 4 and 4. And of course, I repeat it again, everything that you will see here in this part of the graph is not considered and is removed, because that is an error. You don't expect to find anything there. So yes, this is more or less what it is regarding the network. And of course, you can then also export the information. You can export this graph. You can export the network. When you export the network, I will say, remember that every time in Perseus, we export TSV files. So use the specific command in R or Python to import TSV.
So coming back to the issue of some interactions maybe not being as significant as others, you mentioned you could use the profile plot to check those interactions further to see if they if, as you said, they were non-specific binders but they're just lots, or if they're maybe specific binders but there's not lots. So when creating the network, can you tell Perseus to take that into account, or is that something that you have to do manually?
No, you have to do that manually. So you go in the step before, you do the profile plot. You remember yesterday Juan showed you like with the profile plot, and you can calculate the correlation with the others. You do the same thing. And then you see the correlation value. And then if you say, okay, this is not good enough, you remove that row.
And so then can you go back into the networks and delete interactions then? Or
That yes.
okay.
That yes. Yeah. You can also do from the network, but you have to specifically select the node and the edge that you want to remove. Remember that when you do networks, there are 2 matrices. One is node, one is edges. You have to take care of both of them. When you remove a node, you have to then also to go into edges and remove all the edges that relate to that node. Otherwise, you will get an error.
Thank you.
A question here?
You have another here, right? You wanted to ask something? Okay.
My question is about the use of the complement as the negative control. I can see why that works very well when you are using like for proteome-wide interactome studies. Would that perform as well for more like targeted proteomics? Like, for example, targeted meaning that you have a few baits because would that bias against having like 2 proteins with the same binding partners, and by using the complement when analyzing each bait, you wouldn't see this binding partner?
Well, I would say that if you select a bait, 2 different baits with the same binding domain that binds like the same, then you should find the specific binder in both of the situations. So it shouldn't go against that. But you're right, then it will. Yes.
Because the other sample would be in the complement.
Yes.
When
It
you are
could be.
analyzing one particular one. I don't know. Would that be then when I'm in that situation, maybe not using the complement?
Yeah, depends also how many experiments you're doing. If you see here, like for example, some APC1, APC2, they have similar binding proteins, but yeah, in this case, they had how many? 17 different baits and with 4 replicates each one of those. So but if you have like an experiment with only 2 baits and one other with these 2 baits that have similar similar binding domains, and then you have another one, then in that case I would say yes, it may work against you, the complement. I don't know if you wanted to add something.
No, I totally agree. So I don't think we have like a specific number from which number of baits on it. It's really unproblematic, but yeah. If you have a low number, then definitely one has to watch for this effect.
But in this case, also, if you want, as I said before, I could recommend you to then try, instead of the complement, I did it here for the sake of time, but try to instead plot them against the p histons. and then also increase the number of them. And that's something also I think very nice of the study, that indeed they checked both of the things, the complement for everything, but also they plotted against all the histons, and they increased the number of histons, seeing that also the significance increases. So that was a very, like, also I would say, nice idea to then increase so much the number of controls that you use.
Yeah, I think in the parameter interface for the Hawaii plot, one can actually set specific control groups, right? So I'm not sure what is in the internal version, but then you could actually kind of tailor your control group to the.
You see, the only problem that in order to do that, I should now rename all the categories, because you see that they've been renamed with the P histone 1, 2, 3, 4, 5. Then I should then group them. It takes a bit of time, a bit not straightforward, but it's like only not difficult, only time consuming. But yeah, you can try to group them all together, and maybe start with one grouping, one of these, maybe then 3, 6, and then 8 together, with the same categorical name, and see what changes. And here you can select. If we want, we can also plot everything against one of the base, if you're interested in that specific thing.
One last question here.
Can you please show us how to visualize the imputed values? I saw you had the histogram with just the red bars.
Yes, yeah. Sorry, I went quite quickly through that part because it was done yesterday. But yeah, sorry, we can definitely go there again. So, what here? So here on the top, you have this. This is the matrix represented again, your original matrix, in this case, matrix number 10. That here you have the information as usual, for every condition, the LFQ value, and yeah, all the annotations and stuff. You can select them here and change their colors with these buttons here. But then if you want, you can decide to highlight some of them depending on selection from table. I don't know, I will take this one and I say selection from table, and it shows me, I mean, now it's kind of small, but I hope you can see now that there is a little line, orange line here, that are the proteins that I selected here, and this is selection from table. But if you want to instead select from imputation and see only the imputed value, you go here. There is selection from imputation, and now orange would be the imputed values.